So now we continue with lecture 5, organelles in eukaryotic cells, and we will compare organelles in animals and plant cells first, and then we'll talk about the cell membrane and the cell wall in animal and plant cells. So here in this case what you see is an example of an animal cell, and this animal cell here is surrounded by a cell membrane, and it contains all of those organelles. So what type of organelles do we see within the typical animal cell? We see first of all the most prominently in the nucleus that contains the genetic material and DNA in form of DNA and RNA. Then in close proximity to the nucleus we see this membranous structure here, which is the rough endoplasmic reticulum. It's called rough endoplasmic reticulum because it has these ribosomes on top of it. And these ribosomes are, together with the rough endoplasmic reticulum, are critically important for the synthesis of proteins, for translation. Then we have smooth endoplasmic reticulum, or SER. This would be smooth endoplasmic reticulum here. Smooth endoplasmic reticulum is important for storage of molecules and ions that are very active or reactive within the cells, and in order to prevent them to being reactive, they have to be stored in these membranous compartments so that these processes can be shut down and only turned on when they are really needed during metabolism. So this is one task that the smooth endoplasmic reticulum has. Another one is, for example, in liver cells, the smooth endoplasmic reticulum is involved in detoxification processes. And for that reason, liver cells have a very prominent smooth endoplasmic reticulum. The Golgi apparatus. The Golgi apparatus is important for processing, packaging, and tagging of proteins mainly, but also other molecules. As they are being produced, they are being transported to the Golgi apparatus, and they are being transported throughout the Golgi apparatus, and in this process they are being modified and being tagged and being packaged, and then they can be transported as they get released from the Golgi apparatus. They're getting transported now to their target areas. Then we have the mitochondria. Mitochondria are important for the generation of energy-rich bonds in form of adenosine triphosphate, or ATP, which is critically important for many metabolic processes within the living organism. And this ATP is being produced mainly in mitochondria. Then we have the cytoskeleton, and we said the cytoskeleton consists out of microtubules, intermediate filaments, and actin filaments. And the cytoskeleton plays a role in many things, such as division of cells, such as transport throughout cells, the structural stability of cells. All of these functions are governed by the cytoskeleton. Then we have centrioles, and these are the centrioles. These are these are areas in which microtubules are formed and protrude out of, particularly during cell division. The centrioles are important. And then we have the cell membrane, finally, that surrounds this entire structure and keeps everything, the entire cytoplasm that is contained within this cell and that contains all of those organelles, keeps that together and protects this basic unit of life. So now if we continue to the plant cell here, you can immediately see that the plant cell does not just have a cell membrane, but the plant cell also has a cell wall. Now let's look. Plant cells have most of these organelles that animal cells have in common, with the exception of the centrioles, the microtubule areas. And plant cells also have a similar structure, but here they are called the microtubule organizing centers, or MTOCs. And other than that, plant cells have this rigid cell wall that is often made out of cellulose. And this cell wall gives plants additional protection and sturdiness that plants need in order to live, to grow upright and live in harsher, extreme environments where you have sometimes where it's very dry, other times where it's very hot, and times where it's very cold and chilly and wet. these plants need to weather all of those conditions, and the cell wall just gives them extra sturdiness, extra protection from too much loss of water. And so this is a characteristic that animal cells don't have. Then you also see central vacuoles within plant cells. There are some kind of vacuoles in animal cells as well, and these, for example, are contractile vacuoles in Paramecia, which are protists. But the concept of those vacuoles is very different. Here in plant cells, those vacuoles, the main purpose is, first of all, the storage of water. And if you imagine this, so if this is this big vacuole that's filled with water, and that for that reason puts pressure on the outside of these walls of these cells and allows these plants to grow upright. And you know that when you don't give your plant enough water, those vacuoles shrink because the water content in those vacuoles shrinks or decreases. And for that reason, your plant is no longer as strong and sturdy, and it will start to wilt and fall over. If you give it water again, the vacuoles fill up again, this turgor, this pressure within the cell builds up again, and then your cell is straight and upright again. These vacuoles also store metabolic end products or molecules within them. Then we have the chloroplasts, and these chloroplasts and these membranous structures within the chloroplasts are important for photosynthesis and the conversion of sunlight into energy-rich bonds, mainly glucose. And when we talk about photosynthesis, we'll talk in more detail about those chloroplasts. And then we've talked about the microtubule organizing centers within plant cells that from which the microtubules originate. So now let's talk a little bit about these individual organelles, and we'll start with the plasma membrane and the cell wall within those organisms. Let's go a little bit more into detail on this. So the cell membrane that all organisms have consists out of a phospholipid bilayer, and this is what you can see here. And these are phospholipids. So they have a hydrophilic head part and a hydrophobic tail part. And as you can imagine, when you take oil and water and you mix them, or you try to mix them, you will always see that the oil, which is hydrophobic and does not like water, clumps together and the water stays together. And you have a similar phenomenon here. As you take one of those phospholipid molecules and put it together with another phospholipid molecule, then the hydrophobic tails will align next to each other. And for that reason, you have this double membrane, this characteristic double membrane forming, on the outside of which is the hydrophilic part of those phospholipids, and on the inside of which you have those hydrophobic parts. Basically, the oil part is on the inside and the water part is on the outside of these membranes. And this makes them very efficient structures to prevent molecules from just going in or out. I always compare this with a piece of styrofoam that floats on the surface of water. You can try to push it under the water, but it will always pop up. It doesn't want to be underwater because the styrofoam doesn't bind with water. It's not hydrophilic. And you can imagine the same thing here. If you have a hydrophilic structure and you push it into here, it doesn't want to stay in this hydrophobic area here. And for that reason, molecules don't readily pass these membranes. So if we look a little bit more into detail of these phospholipid molecules, you can see here is the fatty acid tail, and these are these hydrocarbon chains. And you can see here the hydrophilic head part. And this hydrophilic head part has charged groups, so they have positive and negative charges. And for that reason, they like the bipolar character of water molecules, which have this partially positive charge where the hydrogens are, and this partially negative charge where the more electronegative oxygen is. And for that reason, this partially positive and negative likes to be in contact with those positive and negative charges here at these hydrophilic head parts of these phospholipids, whereas you don't have charges here on these hydrocarbon tails, and for that reason water doesn't really bind with those hydrocarbon tails. So the membrane does not just consist out of those phospholipids, but there are a number of other molecules within the membrane. There are proteins either incorporated into the membrane or attached to the membranes. There are sugar molecules attached to the membranes and other molecules. And these proteins have when they go through the membrane, have the hydrophobic amino acids in the center that passes through the double membrane here. And the hydrophilic parts of those proteins are on the outside. We'll talk more about this when we talk about proteins and how they are formed. And this model here is called the fluid mosaic model. And this is basically states how the cell membrane is formed. It's not just this phospholipid bilayer, but it is much more. There are many more proteins and sugars that are involved or incorporated into the membrane. So let's talk about cell walls. Not all organisms have cell walls. We said that animal cells don't have cell walls. Which organisms do? So they are the bacteria, and bacteria, again, prokaryotic organisms. And there are plants, fungi, and protists that have cell walls as well. And here so all of those organisms have the problem that they have to protect themselves against harsh environments. Then they have to live with osmotic pressure, so they have to have an osmotic pressure within the cell and have to maintain homeostasis, and also need to be protected against those harsh environments. And all of those organisms have these conditions where they need to protect themselves because they're not as mobile and also single cells in the case of bacteria. So the material out of which the cell walls are being formed is different for all of those organisms. In bacteria, we've already talked about this, it's peptidoglycans, then lipopolysaccharides and proteins. Then in the case of plant cells, we have cellulose, that's the main component of cell walls in plants, then hemicellulose, pectin, and lignin is a major part in wood. Then in fungi, we have the cell wall made out of chitin. Chitin is a structure that arthropods have in their exoskeleton as well. Then glucan and proteins that are responsible for the formation of cell walls in fungi. And then finally we have the protists, and there's a variety of different components that form the cell wall. There's silica, there's calcium carbonate, and there's cellulose. And calcium carbonate structures are often used also to fight pests such as arthropods, because you can buy this in form of diatomaceous earth in a plant store. And this is a very fine powder that is made from protists. And this powder interferes with the integrity of the exoskeleton in arthropods or insects, for example. And for that reason is a very effective ant eliminator, for example. So if we look into the cell wall here, so if this is our plant cell here, and we look into the cell wall, we also have so-called secondary cell walls in plant cells, and you can find that particularly in plants that grow wooden structures, so grow woody stems. And what you will find is that the secondary cell wall, so the cells start to secrete these molecules that form the secondary cell wall, and the cytoplasm gets smaller and smaller and smaller, and the secondary cell wall gets bigger and bigger and bigger, and this is exactly what happens when a tree grows. And so these molecules, in the case of the tree, are the lignin molecules that form the cell wall. Ultimately, the cytoplasm of the cell dies, and then the tree just forms this wooden structure. And every year when it grows more, then you can see one more of these rings in these typical age rings within the trees, and the outer rings are usually the oldest ones, whereas the inner rings are the ones that have been formed most recently. And this is these cells produce these molecules and then secrete them to the outside, form the secondary cell wall, and then the cytoplasm ultimately dies off, and these cells ultimately die off. So this is as far as I wanted to take it with regard to cell wall and cell membrane.